we will first go into Roche's 454 GS FLX machine. This is the base of next generation sequencing. As I said, we should study the base such as Maxim Gilbert sequencing. Still it is not used today, but we need to study what happened when Maxim Gilbert developed a sequencing technology. So like that, this machine is not used today, but still we need to understand what happens during this Roche's 454 GS FLX in this machine. I think you all have heard about pyrosequencing, all these things, terminologies. So this pyrosequencing, all these terminologies first come with this 454 sequencing. So these are the parameters which included in the 454 sequencing. I will just come to that. So we don't need to go for a shotgun sequencing. We can go for a next generation sequencing, which is faster than a shotgun sequencing. And here what we do is that it's somewhat similar to the shotgun sequencing what we are doing. Here also we can only sequence, that's why I need to come back. We can only sequence less than 400 base pair. We have seen in the case of Sanger sequencing, we can sequence up to 900 base pair. Here more the fragment size should be smaller than 400 base pair. In the case of Illumina, it's 150 base pair. So we we broken down the DNA into small pieces than the fragments which we seen in the shotgun sequencing using Sanger's method. So first what we do is that emulsion PCR, I will come to that. So first process in any next gen sequencing technology is the library preparation. So what the library preparation is the first step in next generation sequencing. Actually, we have seen that in the Sanger sequencing, we can for one fold sequencing it will take you can see, we have seen for one fold sequencing it will take 1000 days. So here what we do is that we will first take the genomic DNA and break down into number of pieces, random pieces. What we do is that each piece we will amplify using PCR, and each piece we will amplify using PCR. These steps are the part of the library preparation. Each piece we will amplify using PCR, and each of this bundle of DNA will be sequenced parallelly. Each of this bundle of DNA will be sequenced parallelly. So you know that these after DNA PCR reaction, here there will be 1000 of copies of this fragment, the first fragment we are given. So like that each amplified product there will be 1000 of DNA fragments. So a massive amount of DNA will be sequenced. A massive amount of DNA will be sequenced. At the same time, all this DNA will be sequenced at the same time parallelly in next gen sequencing. So next gen sequencing is usually called massive parallel sequencing. So we first break down this DNA to number of small pieces, and these small pieces we will amplify it again using PCR. That process is called library preparation. Then we will take this library, all we will amplify this library. Each fragments, amplified fragments in this library will be sequenced. So a single fragment will be sequenced thousands of times, so that the result will be, or the sequence will be accurate enough, and as it is done 1000 times or that much times it is will be accurate enough. And from after this amplification we will go for the sequencing in the sequencing machine. So this method is called massive parallel sequencing. So what we are going to do is that 17 people are there. Okay. What we are going to do is that we are going for the library preparation. Okay. For 454, the Roche's 454 machine. So different machines have different methods of sequencing, different methods of library preparation, everything. So first process is, where is my cursor, is our genomic DNA we have taken, the fragmentation of the DNA. First process is the fragmentation of the DNA. The library is first fragmented either by sonication. We can, the genomic DNA is broken down either by sonication, by sound waves, or nebulization. We can force the DNA through a small hole, so that we will get a fragment size ranges from 300 to 800 base pair. A smaller fragment we will get around 400, 300, 400 base pair only can be sequenced by this machine. So we will get the fragments of DNA. Okay. Then we need to amplify this fragment. For amplifying any fragment using PCR, you require a primer. Then only the DNA polymerase can extend and amplify the DNA. So here what happen is that we have broken down the DNA into number of pieces. We don't know what primer to use, because it is randomly this all this DNA is broken down into number of pieces, and what primer need to give you to amplify all these fragments? No idea. For that, what we have done is that we will, for all pieces of all fragments of DNA, we will attach a non-DNA segment in both ends of the fragments. In both ends of the fragment, we will attach a non-DNA which can pair with the primer. So we know the sequence of this non-sequence. They are called adaptase. So adaptase are ligated onto the DNA fragment. So we will add a non-sequence to both sides of the each fragment, every fragment. Same adapter we can give. Okay, and it allows the strand to bind to the emulsion bead. Bead is coming. So we added the adaptase to the both sides of the whole fragment, which is formed from this genomic DNA. Done. Then the 454 sequencing is done the library preparation is done using emulsion PCR. The PCR reaction done here is called emulsion PCR. So for that, we in 454 machine, what we have is that we have a nanometer, micrometer size, not nano, it's very small micrometer sized beads. And these beads surface, I will draw it big. These beads, surface of these beads is attached to the surface of the bead. Lot of primers are attached to the surface of the bead. These primers are complementary to the adapter sequence. We can see a lot of primers attached to the surface of these beads, and these primers are complementary to the adapter which we have added to the both ends of the fragments of the DNA. So, we have beads to which it is the primers are attached, and so then we have taken the beads in a Eppendorf tube, and we will add our adapter ligated DNA fragments. So, the adapter ligated DNA fragments will bind to these beads by base pairing with the adapter and the primer present in the surface of the bead. So, the adapter will base pair with the primer present in the surface of the bead. So, now each bead will be attached to a DNA fragment. So, next is we will denature, we are doing PCR reaction here. So, we will denature, these are double stranded now. So, this double stranded DNA is being denatured by applying 95-degree nail. So, we will denature it, so that double stranded DNA is converted to single stranded DNA. And formation of clonal bead population. Next step is the formation of clonal bead population. So, each bead is coated with a chemical called streptavidin. Make each bead resistant to organic solvents, denaturants, detergents, so that it makes the bead stable. Over billions of bead is used with the primer that matches to the adapter attached earlier. Earlier itself, in billions of beads are there in Eppendorf tube, and these beads will be coated with the primer earlier itself, so that the adapter attached DNA fragment can directly base pair with the primer and get attached to the bead. Each bead is, we have taken the bead in the Eppendorf tube with attached to the DNA fragment. Then we will add oil to this and mix it together, so that when oil and water mix it together, we will get emulsion like this. When oil and water mix together, we will get emulsion like this. And in this emulsion, we can see the droplets. In each droplet will enclose a bead inside them. So, what happens is that when the emulsion form, each droplet will enclose a bead inside them. And this droplet, I think you all people done PCR. So, this droplet can act as a PCR tube, so that these droplets, before we add the oil, I have not said that, before we add the oil, we will add DNTPs, DNA polymerase, water, Mg2 plus ions, all to this. Then only we will add oil, and they mix together, so that it forms these emulsion droplets. So that during that process, each droplet will contain all components needed for a PCR reaction, and it prevents this bead from escaping from this droplet, so that this acts as a micro reactor where the DNA, what the PCR amplification can be done inside this droplet. This can be compared to the PCR tube. So you can see here. So emulsion based clonal amplification, when we have taken the whole PCR mixture, everything here, and then we add oil. So when we add oil, the oil form droplets, and each droplet encloses a bead which contain the DNA attached. This DNA will then get amplified by PCR inside this droplet, and after amplification, what happens is that one adapter is attached here after amplification. Okay, after amplification, again next step is the denaturation for PCR, and one of the fragment will move out, and then it will base pair with the adjacent primer. So like that, again the synthesis occur. Here denaturation occur. One of these fragment will attach to the nearest primer. Again it will amplify, and one of the fragment will move and bind to the nearest primer, and again it will amplify, and the sequencing of the amplification occur here. Again the denaturation occur. The denatured fragment will bind to the adjacent primer, and it will get amplified. Like this, after the amplification, the whole bead will be covered with the amplified product. That is a clonal amplification, and then we will break open the this emulsion droplet, and we will take this bead containing the which is full of the amplified DNA. We will take this bead to the sequencing chamber, so where we will do the sequencing. Up to this is the library preparation method, library preparation step. After this, we will go to the real sequencing technology. So the amplification of the DNA fragment which we need to sequencing is referred to as the library preparation. So in the library preparation, the initial step is the fragmentation of the DNA, and the fragmented DNA will be attached to the adaptase of which we know the sequence already, and then we will denature the DNA into single strand, this DNA into single strand, and this DNA will then we will mix it with oil, so that the emulsion will be formed, emulsion droplet will be formed, and inside the emulsion droplet the amplification of the DNA will be occurring. After each amplification, the amplified product will attach to the nearby primer and continue its amplification, so that after n number of cycles, the whole bead will be covered with the multiple copies of the particular fragment which is already attached first. So our bead will be full of you can see how the emulsion occur. When we mix it with the oil and mix it, then the emulsion can occur. So then we already said PCR, the emulsion PCR amplifies the DNA strand within the emulsion beads. So within the droplets, PCR is conducted. So PCR occur in that droplet. Emulsion PCR, where is yes, here the bead So the bead will be emulsified with the water in oil droplet. So it will be, the bead will be inside a water and oil droplet, and inside the droplet we can get all PCR reaction mixture, including DNA polymerase, primers, buffers, DNTPs, etc. So that inside each droplet it acts as a microreactor where the synthesis of or the synthesis of amplification of DNA will occur inside the droplet. So the whole process of denaturation, annealing and extension will be occurring inside each droplet. So millions of copies of the particular fragment will be amplified, and they will be attached to the surface of each bead. So each bead at first only there was in each bead before amplification it was like this only, one bead, lot of primer, and to one primer a DNA is attached. So after amplification, what it will be the condition? After amplification, the whole bead will be covered with the amplified product. So like that. So millions of copies of the target will be attached to the bead surface. Now the water in oil droplet is 1 micrometer. It's very small. This droplets emulsion droplets size. So that is about the library preparation in the case of 454, the next generation sequencing. So here next is emulsion breaking. After the amplification, we should break the emulsion by using chemicals such as isopropanol and detergent buffer. We can use isopropanol and detergent buffer so that the emulsion can be broken like this. Emulsion, we use isopropanol and detergent buffer so that the emulsion can be broken and the bead can be taken out for the sequencing. And the solution is then vortex and centrifuge and magnetically separated, resulting in a solution in a suspension of empty clonal, non-clonal beads. So we can remove the emulsion and we can take out the beads containing beads which is covered with the amplified fragments. Usually we use magnetic beads for separation. We are not much into going into that. We usually after amplification we usually have beads with amplified DNA, beads without amplified DNA, and we use enrichment bead to isolate the magnetic beads, enrichment bead which will attach only to the, this enrichment bead will attach only to the amplified DNA containing beads, and these enrichment bead are magnetic in nature. So if we add a magnet, these enrichment beads which contain the amplified DNA only get attached to the magnet, and we can separate using magnetic method. So we can separate. Usually when we do a reaction, to every beads the DNA will not attach. So some beads the DNA will attach, some beads the DNA will not attach. Some beads the DNA attached also, sometimes the amplification may not occur. So the beads with contain full amplified product can be separated from the beads without amplified product by binding by using enrichment beads. Enrichment beads selectively bind to the beads which are amplified only. These enrichment beads are magnetic in nature, so they can be easily collected from the non-amplified beads by using a magnet. So bead, everything is done. So I hope you can see a video. So what is pyrosequencing? Usually here the sequencing is not done by dideoxynucleotide termination, but the sequencing is done by sequencing by synthesis only. It is not by dideoxynucleotide synthesis, nucleotide termination. But here also both sequencing technology, either pyrosequencing or Sanger sequencing, both follow sequencing by synthesis using a DNA polymerase enzyme, but it is not by dideoxynucleotide. Here what it happen is that when a nucleotide is added, okay, we need a primer and DNA polymerase. DNA polymerase all time require a 3'OHN to add the next nucleotide. Okay, so when a next nucleotide is added, okay, for example, the nucleotide comes as ATP. As ATP is added, it will be incorporated here as AMP. Okay, and 2-phosphate will be released during this reaction. 2-phosphate plus H plus ion will be released during this reaction, PPI. This phosphate is called pyrophosphate. In pyrosequencing, the sequencing machine detect this pyrophosphate. Sequencing machine detect this pyrophosphate, thereby help in sequencing. So this pyrophosphate is being measured for the sequence during the sequencing. So as we have seen, we have taken the bead into this flow cell, where each bead will be enclosed within these wells. Okay, and then what we do, and in this well we will usually the sequencing by synthesis will have enzyme DNA polymerase. Okay, as I said, apart from DNA polymerase, we add some more enzyme, some more things: ATP sulfurylase enzyme, luciferase enzyme, and apyrase enzyme. Apart from that, 2 substrate also we will add: adenosine 5-phosphosulfate and luciferin. These all things need to be added to these wells. So to these wells we have added the enzymes, and not only the DNA polymerase, but more enzymes and substrate we will add to that each well. And to each well, what we do is that so here is the primer, DNA polymerase is synthesizing. So to each well we will add 1 nucleotide at a time. For each well, first we add ATP. So we can add the sequence like this here: C, T, P. Sorry. You will add the sequence like this: C, T, G, T, C, G, A. This is a sequence we are going to sequence. So next we to that well we add ATP. So the C will be paired by G only. So ATP is added, but ATP can't incorporate here because G only can incorporate here. So no light is coming from that well. Then we add GTP. Then GTP will go here and be here as GMP. It will form triple bond here. So as the GMP bound here, it will release a pyrophosphate. Okay, it will release a pyrophosphate. What this pyrophosphate do is that this pyrophosphate, the ATP sulfurylase enzyme present in the well convert this pyrophosphate to ATP. Okay, in the presence of the substrate adenosine 5-phosphosulfate. We have added all these things here in the well. So this pyrophosphate will be converted to ATP by ATP sulfurylase. These ATP convert luciferin present in the well to oxyluciferin, which gives that light. Okay, we will add each nucleotide, one nucleotide at a time. When ATP is added, it cannot be added here, so the PPI is not produced, so no light came. When GTP is added, it it will base pair with C, and which release the PPI. This PPI will be converted to ATP by ATP sulfurylase, and this ATP will convert luciferin present in the well to oxyluciferin, which give the color. So if GTP is added, which give the color, then we will add TTP. So the next round we will add TTP. It will not give any color. Then we will add CTP, will not give any color. Then we will add ATP. ATP will pair here, and it will convert to AMB and lease PPI, and this PPI will be converted to ATP by ATP sulfurylase, and this ATP will convert the luciferin to oxyluciferin, which give color to the particular well. So that we can flood each nucleotide at one nucleotide at a time to all well, so that we will get signals from all wells parallelly. Some wells we will get the signal, some well we will not get the signal. The wells we will get the signal, it will be A, so we will mark as A from that fragment, so that we can sequence the fragments parallelly. So in the case of this massive parallel sequence. So this is about the 454 sequencing.